This is the first of 3 lectures on the Biomedical Investigations module by Dr. Neil Cross on molecular genetic and cytogenetic methods used in disease diagnosis. So in this session I'm going to cover clinical applications, particularly using cytogenetics. That's looking at chromosomes and chromosome abnormalities, and we're going to focus on prenatal diagnosis, pre-implantation diagnosis, and more generalized disease diagnosis. And then next week we're going to apply these same technologies to a cancer setting. So we're going to be looking at how we can use these genetic technologies to subtype cancers in particular. And we'll start moving on to some variations on PCR technology for genotyping cancers to allow us to subtype them and predict their behaviour. In week 3 we'll move on to some more molecular genetic testing, particularly of tumours, and we're going to look at what we call minimal residual disease testing as part of this. So this involves looking for very rare small numbers of tumour cells that are left behind after therapy has been concluded. So we're going to cover some methods for minimal residual disease testing, and this will encompass some of the methods that I'm going to talk about in the first 2 weeks as well. So this week we're going to focus on cytogenetics, and cytogenetics is looking at chromosomes or looking at the whole nucleus and identifying particular parts of chromosomes. So after this particular session we'll be looking at the principles of chromosome analysis, which we call karyotyping, and we'll also be looking at fluorescence in situ hybridization, which allows us to look at a single specific chromosome or a range of chromosomes within the nucleus of a cell without having to be able to physically see each individual chromosome that we need to do for karyotyping. And in this current session we're going to look at how diseases are diagnosed, particularly chromosomal diseases are diagnosed in fetal tissue, so prenatal diagnosis, a little bit on pre-implantation diagnosis, and look at some of the common chromosome aberrations and how these are detected using cytogenetics and molecular cytogenetics, which is what FISH would come under. So firstly, everyone will have seen a karyotype. This is the chromosome, the constitutional chromosomes within a patient's cells, and this is a male, 46 chromosomes with an X and a Y chromosome there, and these banding patterns are created during the process of karyotyping, which I'm going to go through on the next slide. We call this Gbanding, and basically every single chromosome arm has a specific banding pattern. So we get chromosomes. I'll explain how we get the chromosomes into this configuration on the next slide, but we can apply a very weak trypsin solution to the chromosomes and then a particular stain, usually Leishman's stain, and that will selectively stain up regions of chromosomes with different banding patterns. It's almost like a barcode, and each individual chromosome has got a different banding pattern, and that allows you to identify the chromosomes, and then any deviation from this normal series of bands, and each one of these bands has got a specific number that defines what it is. Any deviation from that normal pattern will be a chromosome aberration, and any deviation from the normal number of chromosomes would also be a chromosome aberration. Now crucially, this must be performed on cultured cells, as we'll see on the next slide. So obviously chromosomes don't line themselves up nicely as in here. This has been done in a Photoshop style piece of software that we use. To do karyotyping we need what we call a metaphase spread, and a metaphase spread is getting cells into metaphase of the cell cycle. So remember, cells go through G1 of the cell cycle where they respond to growth factors, S phase where they duplicate their DNA, S DNA synthesis phase, G2 phase, and then M phase. And within M phase there's prophase, metaphase, anaphase and telophase, and metaphase is the point of the cell cycle where all the chromosomes are lined up on the mitotic spindle, and they have this classic view of chromosomes with a P arm and a Q arm. And at very late metaphase you'll see them as more X-shaped chromosomes, whereas here the two chromatids are stuck together. Now to do cytogenetic analysis or karyotyping we have to get the cells into this configuration, and that involves growing cells. So you can only do this technique on live living cells. So we have to grow cells in cell culture, so that's growing them in cell culture flasks with growth medium, and we need to arrest the cells at metaphase, and to do this we use colchicine, which is a mitotic spindle inhibitor. So this stops the mitotic spindle from separating the two chromatids at anaphase. So we get cells that are stuck in metaphase. The longer we leave them in colchicine, the more condensed the chromosomes get. And when we visually inspect cells that have been treated with colchicine, you will see them rounded up and slightly detaching from the plastic surface that they're growing on, and that's a sign that we've got cells that are ready to divide, but they can't divide because of the colchicine that's been added to them. At that point, we put the cells into a hypotonic solution, so a very weak potassium chloride solution that causes the cells to swell up, and then we fix them in methanol and acetic acid, and that will push all of the water out of the cells, permeabilize and weaken the membranes, and then we take that cell suspension and drop it onto a cold, wet microscope slide. And as that droplet of cells hits the microscope slides, the cells burst, and the chromatin, the chromosomes, stick to the glass. So we're very, very lucky that chromosomes stick to glass very, very well. So the chromosomes are stuck to the glass here, and all the chromosomes, as they hit the glass, will just sort of travel a little bit and then stick to the glass. Then this is what we call a metaphase spread. We then perform a brief trypsin digestion. So trypsin eats away at some of the histone proteins of chromatin, and depending on how condensed the chromatin is, there is two different types of chromatin, heterochromatin and euchromatin. One is more dense, heterochromatin is more dense, and that gives the differential trypsinization and differential staining pattern that we see that give these characteristic bands. All you then have to do is rearrange all of these chromosomes by digitally cutting them out, aligning them as a karyotype like this, and then check every single individual band to see if everything is all correct. And this is what a clinical cytogeneticist would do. Look at patients' chromosomes looking for abnormalities. And whilst they're doing that, they will be looking at each individual band and looking for every single named band. So this is all of the different chromosomes and the naming of the bands on them. You don't need to know this. You've got a P arm. What you do need to know, there's a P arm and a Q arm, and then each band has got its own individual band. So for example, this band up here is chromosome 3P, it's a P arm, and that's 3P25, that little black band there. So every chromosome band has got its own name. And depending on how long you've left your cells in colchicine for, you can either get very condensed chromosomes with very poor resolution, or very long chromosomes, less condensed with much finer resolution. So this is the sort of resolution you get from some tumour cells grown in standard cell culture, whereas this is the sort of resolution you might get from white blood cells that have been stimulated to grow. They grow incredibly fast, and you get beautiful long chromosomes out of those. So we'll start looking at some chromosome abnormalities where cytogenetics is historically been crucial for the diagnosis. The first one, DiGeorge syndrome. It's a relatively common chromosome abnormality, 1 in 4,000 live births, a very variable phenotype because it's caused by an interstitial deletion of part of chromosome 22, and this is a DiGeorge syndrome critical region that contains a key gene here, but then if a deletion is much bigger in one patient than another, you might get different phenotypes. So the phenotype can vary from congenital heart disease, some learning disabilities, in some cases, but not all. So it's due to typically a 3 million base pair deletion of chromosome 22. Can be bigger, can be smaller, and this gene here is rather important. Now most cases are what we call de novo. That means that they are de novo means new. So this means that the mutation or the deletion is not present in either parent, but what's happened is one of the gametes has acquired an interstitial deletion of a chromosome, and then when that is fertilizes, if that's in a sperm and that fertilizes an egg, then the fetus will develop DiGeorge syndrome if it lacks this particular chromosomal region. About 10% are hereditary, and that's because the features are quite variable. Sometimes they're very, very mild, so you can have adults who are known DiGeorge syndrome carriers they have mild symptoms of the disease, and therefore there's a 50% chance that their offspring will also inherit this chromosome with this deletion. And initially and historically it was diagnosed using a karyotype, and this is right on the limit of cytogenetics. It's 3 million base pairs of DNA which is lost, but 3 million base pairs is at the limit of cytogenetics. So these are two DiGeorge syndrome chromosome 22s. This one looks a little bit stubbier than this one. A trained cytogeneticist will look at that, and if you see this in lots of different cells from the same patient, you'll think, yeah, this little bit here is a little bit bigger than this bit here. On a better quality prep, you can see there's a band that matches, those two bands match those two, this black one here matches this, and this white band here, well, it looks a bit bigger there. And that's where this little subband is present. And this is the DiGeorge syndrome critical region band of 22q11.2. So that's what a clinical cytogeneticist would be looking for. You need a really good eye for detail to do this particular job. Now, because that is a really poor resolution of 3 million base pairs, there are higher resolution techniques that can see much smaller genetic abnormalities, and one of those is fluorescence in situ hybridization. Now, it is fairly clear that if we have a karyotype where a chromosome is lost or gained, then this should be instantly obvious to any cytogeneticist. If there's an extra chromosome 21, Down syndrome, additional copies of 13 or 18, Patau syndrome, there are various other chromosome abnormalities, sex chromosome abnormalities as well. So these are very, very easy to spot by cytogenetics, but the small deletions are much more difficult to see, and that's where FISH is useful. So FISH, or fluorescence in situ hybridization, is a technique to add fluorescent probes to either cells or chromosomes to visualize what specific chromosomal regions are present or absent. So this illustrates a cell at interphase. This illustrates a metaphase cell. So this is like one that could be karyotyped. This is at interphase. And this shows staining of a nucleus as it goes through interphase, prophase, prometaphase, metaphase. You can just about make out these individual chromosomes there. Anaphase and then telophase, and then we get cytokinesis with these two nuclei being separated. So wherever the nucleus looks like a fuzzy blob, like this, we call this interphase FISH. Wherever you've got cultured cells with distinct chromosomes that have been arrested at metaphase, we call this metaphase FISH. So if we want to investigate what chromosomes are present here, if we're looking for a specific chromosomal region, we can use some FISH probes, and probes are DNA sequences which match the target sequence. In this case, we're looking at chromosome 7, so D7S 486. It's just a location on chromosome 7. There are various different naming systems for locations on chromosomes. You can have bands, and then you can have these. This represents a dinucleotide repeat polymorphism, but that's not really interesting at this point. And then here we've got the elastin gene for a particular genetic condition. And what we can see, what we've done here is hybridized a green fluorescent probe onto these chromosomes to find out where it sticks, where it's homologous. And the green probe binds here and here on chromosome 7, and the red probe binds to the elastin gene, which is here on chromosome 7. And on this chromosome, there's no red probe bound because in this particular chromosome, the elastin gene has been deleted, another interstitial deletion. So this is how we can use FISH to probe chromosomes, and if we know what we're looking for, we can buy a probe for every single region of the genome and ask the question, is there 2 copies of the gene or 1 copy of the gene? And you can see the metaphase FISH, 2 copies of chromosome 7, but only 1 copy of the elastin gene, same in an adjacent interphase cell. Now in FISH we always use DAPI, which is a DNA binding dye. It just binds to DNA and stains chromatin blue. We use it as a counter stain, so that when we see a green spot there, there and there, it has to be contained within a nucleus, and when we see a green spot here, it has to be contained within a chromosome. So we call that a counter stain. It binds to chromatin. So this is how FISH works, and there's bits on here which are a bit more complicated than what you need to know at the moment, but we start off with what we call a purified DNA clone. This is just a piece of DNA that we know represents the region of interest that we are looking for. So for example, in this case, the elastin gene. So we can purchase off the shelf a purified DNA clone, which will contain usually a large plasmid that contains an insert with the entire elastin gene, or just part of the elastin gene within it. So we can buy that. We can label it either red or green. So we can buy nucleotides that are labeled with fluorescein isothiocyanate, or FITC, which is green, or we can have them labeled with Texas red, which is red, and there's loads of different colours in between: spectrum green, spectrum orange, spectrum red. So you can get any colour that you want. Okay, so we've made our label probe by mixing the DNA, this purified DNA clone that we can just buy. We can label that with nucleotides by mixing the red labelled nucleotides with DNA polymerase. We'll label our probe, and then we hybridize this probe onto a chromosome prep on a microscope slide, and that can be a metaphase spread like I showed you earlier, or it could be a piece of tissue. It could be a piece of formalin fixed paraffin embedded tissue that has been put onto a microscope slide, particularly if we're dealing with cancer, for example. But we have either cells or chromosomes on the microscope slide. We hybridize the probe, having denatured the DNA first, allow the probes to anneal, wash off the excess, and then we excite the microscope slide with specific wavelengths of light that excite those fluorochromes. And if we excite with 530 nanometer light, we'll be able to see red light emitted. If we excite with 450 nanometer light, which is more in the blue, we'll be able to see green light emitted. And if we excite with UV light, we'll be able to see blue light emitted. All we then do is we look at the chromosomes and we see usually red spots, green spots, maybe on a blue chromosomal background. So the key points here: if you're going to do metaphase FISH, it must be performed on cells which you have grown in the lab and then arrested them in metaphase and put them on the glass slide. Or you can do interphase FISH, which are cells which are not necessarily dividing, or you could do that on pathology material, so a formalin fixed paraffin embedded tissue section is perfectly good for using FISH on. And this little animation shows the whole process. So here's some chromosomes stuck to a glass slide. So chromosomes are denatured to single-stranded DNA. We normally put them into formamide at 70 degrees. We'll label the probes. So here's some probes, some DNA. This is some unlabeled DNA. We'll label it red. We'll denature the probe. So we've got red and green, and then we've got unlabeled as well. And we will hybridize those onto the chromosomes. Where they match base for base, they will hybridize. The rest will be washed away. And then we will label those chromosomes with DAPI. So the chromosomes are blue, and then we're looking down on them from this angle, and we'll see a red spot there, and a green spot there. And it'll give us an image something like this. So what we can now see is how much simpler DiGeorge syndrome diagnosis is with FISH instead of cytogenetics. So if we look at those chromosomes that we saw before, there's chromosome 22 with a little deletion there that you can barely see. What we can do is buy FISH probes that will bind entirely within the DiGeorge syndrome critical region here in red, and somewhere else on chromosome 22 in green, hybridize them onto some cells from patient, and patient A has only got one red spot, two green spots, whereas patient B has got two red spots, two green spots, and on this alone we'll be able to say that that is a DiGeorge syndrome patient. We'd often use this to confirm observations by cytogenetics, or to confirm the phenotype of a newborn that has symptoms of the George syndrome, and we can do a quick FISH test to be absolutely sure that it is the George syndrome, because this is one of those diseases where very quick treatment actually can have a very favourable outcome in some patients. Now we can also do more complicated versions of FISH. We call this M-FISH, or multicolor FISH, or spectral karyotyping. And in spectral karyotyping, instead of just having one green probe and one red probe, like we've seen in the previous example, we can purchase, we won't do it ourselves, we can purchase chromosomes which have been labelled with specific coloured nucleotides. So there are at least 24 different fluorescent fluorophores that we can attach to nucleotides, which means that we can do 24 colour FISH, because there are 24 different chromosomes, chromosome 1 to 22, X and Y. And if you can have each chromosome labelled in a different colour, you can see exactly where each individual chromosome is. And this is really useful for spotting very cryptic translocations between one chromosome and another. Now what you can see is you can't do spectral karyotyping in an interphase cell, it's got to be on a good quality metaphase. Okay, so to see if you understand all of this, here is a whole series of things that are done in terms of FISH, and this is a list of things of why they are done. They're all in the wrong order, and the explanation is not linked here. So what I want you to do is just take a few minutes out, read through these, and order them in order from 1 to 9 or 10, however many there are, and try and link up what is done to the other half of the table. And if you can do that, that will confirm that you at least understand FISH. Now a few parts of that previous task might have been slightly difficult because there's one area that I've not yet told you about, which is the fluorochrome labels themselves. So you may well have got to this question, this bit here, take 3 independent images using a monochrome camera, and the bit I'm going to tell you now is going to make that make more sense. So the fluorochrome labels, the ones that you need to know about, I would stick with FITC, which is fluorescein isothiocyanate, which is green, and Texas red, which is red. They're the two easiest ones to remember. FITC, green, Texas red is red. But there are lots of others as well. There's Psi3, Psi5, a whole range of them. And they all have very conjugated structures, so that's alternating double and single bonds, which is classic for a fluorescent molecule. Look for any fluorescent molecule, ethidium bromide, FITC, Texas red, prapidium iodide. They all have a conjugated system, often based around alternate double bonds and ring structures. So the key thing about all of these is they absorb light at one wavelength and emit light at another wavelength. So for example, Psi3 absorbs light with an absorption maximum here and emits at a longer wavelength. Let's say Psi5 here, absorbs here in the orange and emits further into the red. So all fluorescent molecules absorb at one wavelength and emit at a longer wavelength. And we can see this in more detail here, and it helps explain how a fluorescent microscope works. This is the absorption spectrum for something like FITC, fluorescein isothiocyanate. It will absorb light in the blue spectrum, so you use a blue light to excite FITC, but it will emit in the green in the green spectrum. So the absorption maximum might be 420, 430, maybe probably more like 450 nanometers, and it will emit with an emission maximum above 500, maybe 520, 530. If you look at those two spectra side by side, there's a bit of overlap. So what we want to do is excite with blue light and just observe the green light. And the way we can do that is we use fluorescent filters within the microscope. So here's a different example. This is something like DAPI, which emits in the blue. We can have an excitation filter that allows UV light to hit the sample, and then we have a dichromatic mirror which then only allows the blue light to go to the eyepiece. So you shine ultraviolet light, or either violet or ultraviolet light, onto the sample, a very short wavelength. That will hit the sample. Blue light will be emitted, but you will only allow the blue light out of the up to the eyepiece or to the camera. So everything beyond this cutoff here at 400 nanometers, anything below 400 nanometers doesn't get to the eyepiece. Everything above 400 nanometers does get to the eyepiece, and you can separate the excitation light with the emitted light. So we can do that with filters. We can have excitation here with blue light. We can have a filter, say somewhere in between here, and then we can observe the green light. We can excite with blue and we observe green light. And there's a term that we use here called a Stokes shift, which is the difference between the excitation peak and the emission peak. And a large Stokes shift is sometimes very, very preferable because you don't get overlap between the excitation and the emitted light. So this is just summarised here. Texas red absorbs at 520-ish, emits at around 600. You have a cut-off somewhere in between so that the green light that you're shining on the sample doesn't get to your eyepiece. You only see the emitted light in the 600 range, and that allows you to see, even though you've got a green light shining on the sample, you only see the red emitted light. Now if you click on this link here, it will, from the original PowerPoint slides, it will take you to the Olympus Resource Centre, where you can look at lots of different filters for different fluorophores. There are hundreds of fluorophores to choose from, and most of them have got their dedicated filters for fluorescent microscopes. So what I'm going to do now is move on to how prenatal genetic diagnosis is performed and how we can use either karyotyping or fluorescence in situ hybridization to diagnose genetic abnormalities in developing fetuses. So firstly, before you do any genetic analysis of fetuses, you need to obtain the genetic material. And there are various different ways of doing this. Most people have heard of an amniocentesis, where a needle is inserted and a sample of amniotic fluid is removed, and we tend to do that beyond week 15. I'll come back to this in a moment. There is also a chorionic villus biopsy, which is sampling of material from the placenta, and the placenta is genetically fetal, not genetically maternal. And so you can either go via the transcervical route or the transabdominal route to take a small biopsy of genetically fetal placental material, and you can use that material to find out what the genotype of the fetus is. Now this would only be done if there was a good reason for doing it, because this can induce a very small chance of spontaneous miscarriage. So if the fetus was suspected Down syndrome due to other biochemical markers or something on the scan that was abnormal, then a chorionic villus might be performed to then perform genetic testing on the fetus. And this is summarised here. We might want to look for unknown, unanticipated genetic abnormalities, chromosome abnormalities. So if a fetus is not developing properly, we can take a chorionic villus biopsy and do full chromosome analysis. Or if on the scan suggests that fetus is developing characteristic for Down syndrome, we would then take a sample and look for additional copies of chromosome 21 by FISH or karyotyping, almost certainly now by FISH. And if the fetus is at risk of a Mendelian inherited genetic disease that's caused by a specific mutation, something that we know about in the parents, such as like Duchenne muscular dystrophy or Huntington's disease, we can take out a piece of this genetic material and perform polymerase chain reaction for a specific mutation, but we'll cover that, or you will have covered that in other lectures. So we can look for some common chromosome abnormalities in developing fetuses, and this is quite often taken from an amniocentesis, as shown here. And what we do, typically use a 5 or 6 probe system looking for chromosome 13. If you have an additional copy of chromosome 13, it's Patau syndrome. Additional copies of chromosome 18 is Edwards. Plus 21 is Downs. And then we can look for plus or minus various sex chromosomes. Here is a metaphase spread of some fetal material, and here is an interphase spread, not an interphase, an interphase cell from the same patient. And you can just go through there and say, look, there's 2 blue spots, there's 2 pinky purple spots, there's 1 green. Okay, that gives us it's male. There is 1 red, so it's XY, and it is normal for all of the common chromosome abnormalities. So this would be the result that if this was being done for looking for Down syndrome or one of these other chromosome abnormalities, this would come back as completely negative. This is what you'd expect to see in a normal pregnancy, both at interphase and at metaphase as well. So prenatal genetic diagnosis tends to be done when we either see something abnormal on the scan, or we know that there is an abnormality potentially present, such as likelihood of carrying a genetic disorder, and we would need to access the genetic material. And if that genetic material proves that the fetus has an abnormality, it is then entirely up to parental choice as to whether that pregnancy continues or is terminated. Now, if you know that there is a genetic abnormality present, such as one or both parents carry a genetic abnormality, and conceiving naturally and then aborting fetuses with a genetic abnormality doesn't fit quite right with those parents, then there is a way around this, and that is to perform pre-implantation genetic diagnosis. So this is done if the parents are carrying a genetic disease and we know the exact mutation. And in this process, we perform in vitro fertilization, so harvest eggs from the mother, fertilize those eggs with sperm from the father, allow those to develop to the 8-cell stage, and then pluck out a single cell and then perform genetic analysis on that single cell. And if that single cell is normal for the mutations that we're looking for, those developing fetuses are implanted into the mother. If they are genetically abnormal, then they are discarded. So we call that pre-implantation genetic diagnosis. And this is just summarised here. You typically get half a dozen fertilised eggs, get them to the 8-cell stage, and biopsy a single cell out of there and perform genetic analysis on that. We can perform FISH on that if we're looking for a chromosome abnormality, or we can perform polymerase chain reaction to look for a specific mutation. So don't worry too much about polar body here, but basically we are looking at the 8-cell stage. We call it a blastomere biopsy, and we take a single cell out of there and perform genetic analysis on there, or we can leave it to the 16-cell, later than the 8-cell stage, and take more material for analysis, but you've got less time to then implant the developing fetus if the initial tests fail. There are some risks associated with this. There were concerns that this will cause embryo damage. We're losing 1 cell out of 8. Will the fetus develop normally? Well, we know mice develop normally, and we now know that this technology has been used for a few decades now, and we know people that appear to have no long-term health issues for having one of their 8 cells removed early in development. But we haven't got very, very long-term data. But we think it's okay. I'm not sounding that convinced, but there is no reason to suspect that this is a problem. IVF risks. There's a chance of not achieving pregnancy. We know IVF is not the most reliable of techniques. Not everybody who goes through this process gets to pregnancy, and the embryos might not implant. You might get 6 fertilized embryos, 4 of them might have a genetic mutation, the other 2 might not implant. There is a chance of misdiagnosis. We're dealing with a single cell, trying to perform genetic analysis on a single cell, and if you put a single cell in an Eppendorf tube and sneezed somewhere near it, you would transfer more DNA into that tube than the single cell contained. So therefore the risk of contamination, false negatives, are a real problem here, where the developing embryo has got a genetic mutation, but you miss that because lots of normal DNA has contaminated the sample. So you get both false negatives and also some false positives, and also sometimes no result because you're trying to extract DNA from a single cell. So there's a chance of misdiagnosis, but greater than, we're approaching 95% specificity, I believe, now with this. And it's very expensive as well. So this is only done in situations where there is a severe genetic disease and where carrying a child, terminating a fetus after a prenatal diagnosis is not an option. So tends to be offered where if parents have maybe terminated one or two affected fetuses in the past, then these would be higher up the list for getting pre-implantation genetic diagnosis. And if termination is ethically and morally against your beliefs, you're more likely to get pre-implantation genetic diagnosis offered. But it's only for a very specific list of genetic conditions. Okay, and as a bonus for those who've made it all the way to the end of this video, here's a solution to that little problem that I set you earlier. Okay, that's the end of this video, and bring questions that you have on this to the live session next week.